important module for the long non-coding RNA knowledge base is the trait association aspect of long non-coding RNA and eQTL information that can be integrated. And we actually carried out a trait association analysis using summary statistics or using a method called the summary Mendelian randomization analysis to prioritize long non-coding RNA, or to prioritize genes part of the long non-coding RNA across 323 traits in the UK Biobank. The trait association results can be accessed on the LNCRNA-KV knowledge base using the trait association module by clicking on either the link on top here or the main menu. And when you or particularly when a user goes to the trait association results, it actually takes you to a table showing all the traits that we've analyzed. These are traits which we have greater than 5,000, actually greater than 5,000 cases. So we selected these traits from the UK Biobank, which is publicly UK Biobank, which has publicly available summary statistics across these different traits for the different GWASs that they've performed. And we have the eQTL results from the long non-coding RNA, and we try to integrate that to see if we can prioritize genes or long non-coding RNA in these different traits. So the trait page shows this table, which actually has all the trait information across the 323 GWASs or traits. Users can actually click on the phenotype ID across which is provided by the UK Biobank, and that will take you to the actual page for the particular trait in the UK Biobank. As you can see, this trait is snoring, and it gives you all the information, the number of cases, controls, the SNP heritability estimates, and different and other different annotations. And in addition to that, we give the number of cases, controls, the total number of subjects or samples involved in the analysis for that particular trait, and the prevalence calculated by the UK Biobank. And here, by clicking on each individual trait or description, we actually dynamically generate the number of genes that have been prioritized across each tissue with a raw SMR p-value of less than 0.05, so nominal significance. And by clicking on each trait, the bar graph changes, and you can see the number of genes that have been prioritized. So if you were interested in a particular trait, say we were looking at depression, and as you can see here are different traits for depression. And if we just go to the first one, which has a non-cancer illness code, self-reported depression, we can click on the description, and this shows all the tissues for which the SMR results are available, as well as those that have P genes that have been prioritized at PSMR less than 0.05. And for example, if we wanted to go to access the brain results, user can click on any of these bar graphs, and it will take you directly to the brain trait association page. And within that, we display the actual entry that you clicked on for that particular trait, and the results. All the results are displayed here are at P less than SMRP less than 0.05, and you can get the full table of results. You can download them, or you could access them through the web interface and sort them by any of these criterias, by the GWAS p-value, or the SMR p-value, or the actual SNP ID, or the gene ID. Any of these criteria that you can use to identify interesting long non-coding RNA or interesting genes for that particular trait. So for example, if we sort this by SMR p-value, we can see that these genes have been prioritized at the top, and users can actually click on any of these genes of interest and go back to it links back to the gene page, where you get all the information for that particular gene, including the transcripts, again, the expression, the tissue specificity score, the network information, the eQTL information. You can get the genome browser track for that particular gene, and kind of makes a full circle for discovery of genes for that particular trait. And you can actually also see if uh, other tissues have, if you're interested in other tissues that have prioritized genes for this particular trait, you can go back and select the particular tissue, and the results for that tissue will be will be displayed for that particular trait.